membrane, just 2 molecules thick.
It's kind of groovy sounding. All right, so let me show you. This sounds a little scarier.
The membrane of the cell is a bumpy film of fatty molecules. Proteins rise like trees from this bumpy layer to pump and channel minerals and nutrients through the membrane. Other proteins use their branches to trap hormones that activate the cell. Some hormones are small enough to slip through the membrane to instruct the cell to move, grow, divide, or even die. Larger hormones dock at the protein receptors. The receptors send messenger molecules to the enzymes and organelles crowding on the cell.
Okay, so the point of showing you that is that the cell membrane is more than a line that we've drawn on your paper. And that's I think the most, not so much the hardest thing to understand, but it's conceptually not that easy. Well, what we're going to find is that the cell membrane is actually 2 layers thick, like what they said, of those phospholipids. So we're going to draw those in just a minute, and that's what we'll also put in his model. But they also mentioned some proteins and that hormones kind of dock on there, and then it tells the cell what to do. So we're going to find out the cells have to communicate with each other, and that's part of this. So let's draw it, and then we'll look at the communication, and we have like a little virtual, little interactive lab thing we're going to do. So, all right, so the first thing we're going to do is write down the cell membrane structure. So cell membrane structure. And so if we take that line that we've been drawing around, like when you drew your animal cell, or your cheek cell, you just drew that little line. We said, oh, that's the cell membrane, and that's how it looked like. But if we were to look under an electron microscope, not a light microscope, we could actually see these parts. So the first thing we're gonna do, leave you enough space because we're gonna write above your diagram. So I'm going to take my yellow, just because I think of yellow being kind of fat-like, and we're going to draw a phospholipid. We're going to draw 2 layers, but it has a head end and 2 tails, so it's kind of like a balloon with 2 tails. Okay, so you don't have to circle I'm just going to do that so that way you can kind of stand out a little bit better. And then we have another one on the bottom. We're going to draw like 6 of these, 3 on top, 3 on below, like that. Okay, so let's just do 3 of those in a row. All right, so this is a phospholipid. How many layers do you see?
2.
2, so we're going to call it a bilayer. So if you were asked, which you will be, what is an organic molecule that composes the cell membrane, you're going to say, well, phospholipid. And then we're going to put in some protein. So you'll say, well, it's mostly phospholipid and protein. So there's 2 of them. We're going to make it look like a cross section, but we know from the view that you saw, it's more like you're moving on top of it. So it looks like what I usually explain it to be like, you know, the little, it's not like the balloons you can buy on a stick. You know how the balloon just kind of stands there, you know? It's like if you get a bunch of balloons and each balloon is a helium balloon, but it had 2 ribbons, and you have a bunch of them and you're holding them all together. And, if you walk with them, they're going to kind of bounce, right? They're kind of in place because they're not like flying around, but they're still kind of bouncing. That's what your cell membrane does. So right now, when you're looking at it, all you do is see is your skin. But beyond that, if we were to look at it under the microscope, it actually would be moving, which is kind of bizarre. So those are the phospholipids. Here's the honors version of that. The heads are hydrophilic. Okay, so what does hydro mean? Water. And if it's philic versus phobic, is that water loving or water hating? Loving. So water hydrophilic, it's like I feel it, I like it, whatever, I don't know. But so that is actually a hydrophilic head to that phospholipid. And the tails are hydrophobic. So it's wild that they love water here, but gosh, we have to try to get the water through those tails that doesn't like it very much. So we're going to notice that it's a natural movement of water from one side to the other, but it's not so easy sometimes. So these little tails are hydrophobic. So if I ask you what are the two parts of a phospholipid, you can say that there's a hydrophobic or hydrophilic head and hydrophobic tails. Okay, and those are actually fatty acids. So it's like, remember the triglyceride had 3 fatty acids? Well, this is just one with 2 fatty acids, so it's similar. Okay, so there's that. Now take a different color, and we're going to draw a protein, and it doesn't matter what color you use. We're going to draw a protein that actually goes all the way through the membrane. So this is called an integral protein because it actually passes from one side to the other. And on that one of those videos, I think it was the second one, you saw some stuff kind of shooting out of the membrane, and it was probably going through one of these things. So it almost looks like 2 sides of a nose. All right, so this is an integral transport protein. Now, sometimes they call them carrier proteins, but basically this is how it works. Really tiny molecules. Oh, we lost a fatty acid. If it's a really tiny molecule, like, and I don't want to break his model, so let's say it is. All right, so y'all agree that all 3 of these balls are different sizes, right? Mm-hmm. Okay, so when solute molecules, and y'all know molecules in general, so this could be a protein. This would really be cheating. It would be much smaller than this. Let's say this is an amino acid. So amino acids are really tiny. Proteins are a lot bigger. This thing, not going to pass through here at all. It's just going to bounce off. So if you notice in the video, some things just hit the membrane and kind of bounced off. Well, it's because they're too big to get through. Other things might be bigger, but they can get through. It just might take them a while to get through and squeeze through those balloons, okay? So when that happens, we've got these cool transport proteins that can actually act as a passageway, okay? But not because he's actually taped it closed. But this passageway allows it to go through a lot quicker. So it's almost like the 10 items or less line at the grocery store, okay? This one should have passed right on through, okay? So really tiny molecules can pass a lot easier. So that's called solute size. And we're going to talk about that with your lab. Okay, so that's transport protein. Now let's take the color. We're going to draw a couple more of those lipids. All right, and this time we're going to put a different kind of protein on there. He put this little hot dog looking protein up here. He put, notice this yellow one? And what's the difference in this protein and this protein compared to this protein? Other than the color.
Which one?
This one and this one. How do those compare to this one? I know the shape's different. Think about their proximity.
I was gonna say,
Or like their position.
that's
That's
between outside, and that's in the.
Right. This one goes all the way through and passes through. These are just kind of stuck towards the top. They can be on the bottom. So these are called peripheral proteins, on the periphery towards the outside. And what these do is they are going to be active with those receptors, and they're going to have those cool little branchy things that come off of them. So your cell membranes have to be able to communicate with another cell and back and forth. So that's what we're going to draw next. This is a different color, whatever color you have. We're going to make one, and it kind of looks like a tooth or a heart. Yeah. So we're going to draw that and make it like a little shape or something up there. We'll just kind of throw some more in there. Oops. Color those in. Okay, so this protein is actually a peripheral protein, but it's called a receptor. I'll move out of the way. See my little papers. So here it is. So it's kind of stuck in there. And this shape is going to be unique to whatever it binds to. Okay? So if this is, let's say, one of your muscle cells or a stomach cell or a heart cell receptor protein for insulin, if insulin attaches to it, what's this insulin going to tell this cell to do? What's it going to tell to move into the cell because you need it? Sugar. So think about this. If you're diabetic, you're eating carbs or you're drinking carbs, but that glucose will stay in your bloodstream. It stays in there. It doesn't go anywhere until the cells tell it to, hey, you need to start bringing that stuff in here. And so that's what insulin does. It actually will bond. Pancreas spits it out, okay, puts it into the bloodstream. It attaches to the cell, to that protein, and says, okay, you need to start moving that glucose into that cell. Now, where does the glucose go? It's gone out of the bloodstream, thank God. Okay, otherwise, those cells aren't going to have energy, and that person can go into a coma. So we've got to get that glucose out of there, and once the glucose moves into the cell, where's it going to go? What organelle is going to take it? Mitochondria, and now it's going to convert it to what? ATP. So your cells are so dang complex that it's amazing. And people ask me, did life just exist? Makes you wonder. You know, it's pretty complex, isn't it? So let's draw a little hormone. And you can just do it with your pencil or something, but let's draw something that fits that, and I'm going to call it insulin, okay? So here's my hormone.
You love biology, don't you?
I'm a biology major, yeah. So I wasn't a teacher major, I was a biology. And then I got my teaching certificate. I wanted to be a teacher my junior year in college, but I didn't want to go longer. So I finished my biology degree first. And then I went back to UNC Charlotte. I went to Chapel Hill and then I went to UNC Charlotte. So. All right. So there's that. Now we got to draw one more little one more little protein and draw your carb ID tag. So do you have one more color left?
We have like.
So I've got 1, 2, 3. Okay. So I've got, we're going to draw another protein like this, but it's not going to have like that funky shape. So I need to get rid of this and draw one like that. I guess you could color that, I guess, but We're gonna do one more protein. That'll be our last one. Okay, and then we're going to draw a carb ID tag. So on that video, do you remember how it had those little branches coming off? Do you remember that? It was in the kind of eerie little video. So what those are is they're glycoproteins, or what we call carb ID tags, okay? And these, there's different purposes for them. There's different receptors and things that are out there, but those a lot of times just mark your cells as your own. So that is a carb ID tag that should be unique to your cells. So that's how your immune system knows what doesn't belong in your body. So if it has different carb ID tags, it's like, alert, alert, and it's going to start rejecting whatever it is. So what happens if you get somebody else's, you need a kidney, and somebody donates you a kidney, what's going to happen? You can reject it. So what do you have to take? You know what you have to do to prevent your body from rejecting it?
Take medicine?
Yeah, it's just a medicine. It's called just an anti-rejection drug. There's different kinds, but you have to take that because and it prevents your body from your immune system from picking up that it's not yours. So that medicine kind of masks that. So it says it's okay, you can have that kidney, but if you don't take that, your body's going to go reject, reject, and it's going to fail. It's going to do what? I can't hear. Yeah. It's okay. So yeah, I mean, that's kind of a neat thing though. So you have your own. So how would the ID tags compare to if you were an identical twin?
They're almost the same.
Should be the same. So that's why you're a perfect organ donor. So you'll have should have the same carb ID tags. Now this is the sad part. And somebody last period actually shared that 2 of their family members have diseases that are considered autoimmune. Okay, so have you ever heard of autoimmune diseases? So one's like lupus, diabetes can be that way. Crohn's disease can kind of be that way. There's different types, but autoimmune disease is basically your body starts to doesn't even recognize its own ID tags. It rejects your body rejects itself. So that's that's kind of sad. I mean, it's hard. And some of those might be genetic. There's a whole lot of kind of stuff there's a lot of research and stuff. Yeah.
Is there a way to get rid of them? My friend mom said she was like, oh, it's gone. Because, you know how they have scars and stuff? It went away. She was like, yeah, it's gone.
There's a lot of treatments for it, but all right, so that's how the cell membranes work. So what we're going to do now is there's a little virtual lab where you guys are going to look at different cell sites, and you're going to introduce different things, stimuli to them, and we're going to see how the cells respond. Okay, so it's the second sheet in your packet. So y'all need to get your computer, and we'll turn to the second sheet. And I haven't forgotten, y'all had some homework. We had to look up something about stem cell research. So, yeah, I'll collect it.
I just saw him
You have to at least write
and I
some.
passed out. So I was like, oh, cool. I'm serious.
We're gonna let folks share it. We're gonna let you share.
I do remember
before
what it
we
was. It was where they take the stem cells.
Okay, get your computer. Don't worry about it right now. Don't tell me. You just need your computer. And we are going to do.
I used to, on my Xbox, you know, I was doing
Is this it?
Yeah.
Which one? This one or this one?
Right here. Yes, ma'am. Yeah. So what you're going to do is you're going to just research one use of stem cells. Like right now, like any type of research that's being done or some cool thing with stem cells.
Can I take mine?
No, you're not going to tell it to you. You got to have one. Then you can share it. Okay. So funny thing, this we can't do. We could last semester, but somebody has purchased or has the rights to this link now, so we can't use it. So we're going to do this part, so we have to ignore that. But what you're going to do is go to Google, and instead of typing in a long URL, we're just going to search. So just go there. And this does have sound, so you can either, but it also has subtitles, so you can read it, or you can plug up and listen, or just turn it really low. Folks did it all different ways. We just don't want all of it blasting, so you can turn it down low, listen, or just read it. Okay. All right. So what you're going to type is this learn genetics, just what it says up here. Okay. I didn't even put the dots. I just put learn genetics Utah, and then I put dropping signals, and it comes right up. Okay. So if you're interested in genetics, this is a great site. There's some really cool stuff that's out there. See, look at that. Like the effect of drugs and like mouse price. Dropping. And it should be the very first thing that comes up. And then you'll have to hit that little Adobe. Piece.
Mine has one of the other two systems.
And then it'll look like this. Okay, so those 5 cells are actually on this side right here. Okay, and on the top, these are the different conditions or chemicals that you're going to expose the cells to. Okay, so we'll do the first one together just so everybody understands, and then you can just go for it. So we're going to end up clicking on the muscle cell first. If I click on that, we'll see the 4 different stimuli that are over here. And we're going to do hormone first just because it's first in our little paper.
My computer's so low.
It's okay. That way you can just watch it. You can see how we do it. So we clicked on the muscle cell, and we're going to click on the hormone first. And these will tell you what they are. So like a hormone, we know is a protein molecule. It says travels through the bloodstream. That's true for a lot of hormones. And then cytokine is actually a growth factor that causes cells to divide. Typically, nitric oxide. Does anybody know what that is? Or nitrogen oxide?
So they put on you to fall asleep.
It's like laughing gas. And then light. Okay.
What do I type in again? I have like learn genetics Utah, but what else?
Right here where it says dropping signals. Just type that in.
Which one are we doing?
Okay, so we're going to click muscle, and we're going to click on the hormone just because it's the first one in the list. I don't know why they put it at the bottom. So you drag it over. And what you can't see
In
is
response to a hormone, this muscle cell is taking up nutrients from a nearby blood vessel.
and that's all we do. So probably they're talking about insulin. So what we'll write is right here. So that's what we actually will put right here, that that it takes up nutrients from the bloodstream.
Mine's not working. It's not letting me click the little flash player thing.
You can, I don't care, if you want to use this one, you can.
Yes, everyone night.
Okay, perfect. This only took about, I don't even think it took them 15 minutes. I think it was closer to 10 minutes, but But anyway, you just go for it. And then if you want to share with somebody, if there's two of you that want to do it together, if your computer's having issues, or you can use mine. Or
What if you're like and then it'll start talking.
Okay.
right, go back. Or I'll tell you what, just go up here.
Yeah.
Next to it. Right, drop it. Drop and say next. There it goes. That'll be the first thing that's going to
In response to a hormone, this muscle cell is taking up nutrients from a nearby blood vessel.
go. Drop incisions, just add that. I have no idea. Oh, now you just click it over. Now you just drag the next thing. Okay. I don't even know if it's turned on. The computer's so jacked up. There we go.
This muscle cell is taking up nutrients from a nearby blood vessel.
All right, anybody else have any questions or any problems with anything?
This muscle cell is responding to a cytokine by dividing to make two cells.
You should like write the notes in the box.
Yeah, just put it right there in the box, and how that would call, what that calls, what response that, whatever that stimulus was that caused. You can.
This muscle cell is responding. Nitric oxide is causing these muscle cells to relax.
What's this?
This one, the bag on the desk. This stuff, right there.
Just put underneath. For now.
In response to visible light, photoreceptors generate a signal. The signal travels to a neighboring cell, which is part of a relay chain that ends in the visual center of the brain. In response to visible light, photoreceptors generate a signal. Signal travels to a neighboring cell, which is part of a relay chain that ends in the visual center of the brain. Nitric oxide is causing this photoreceptor to go through light adaptation. Light adaptation Light adaptation allows your eye to adjust as you move from the dark to a brightly lit room. This photoreceptor does not respond to this particular cytokine. This photoreceptor does not respond to this particular hormone. Ultraviolet light from the sun damages a cell's DNA. DNA damage can result in mutations in genes that control certain behaviors. This case, a mutation is causing a cancer cell to grow and divide better. Ultraviolet light from the sun damages a cell's DNA. DNA damage can result in mutations in genes that control certain behaviors. This case, a mutation is causing a cancer cell to grow and divide very. This skin cancer cell is responding to nitric oxide initiating programmed cell death. Also known as apoptosis, programmed cell death is a form of self-destruction. This skin cancer cell is responding to a cytokine signal by growing and dividing rapidly. This skin cancer cell is responding to a hormone by becoming unstuck from its neighbors and migrating. It will enter a nearby blood vessel and invade another tissue in the body.